Welcome to the AI Research Roundup. I'm Alex. Today we're looking at a paper trending on X, published on February 6, 2026, just over a week ago. It introduces a novel global tokenization approach that drastically improves protein generation, achieving a designability score of 0.87, compared to roughly 0.27 for prior discrete models. The paper is titled Adaptive Protein Tokenization, and as we'll see later, the implications for designing functional proteins are quite significant. Alright, let's start with the core idea illustrated here in Figure 1. The top row shows how traditional protein tokenizers work. They process the structure locally, meaning each token corresponds to a specific spatial neighborhood. You can see the protein being built piece by piece, essentially from one end to the other. By contrast, the bottom row demonstrates the new global approach. Here, tokens don't represent spatial sections. Instead, every token adds detail to the entire structure simultaneously. So the shape starts as a coarse global approximation and becomes increasingly precise as more tokens are added. So Figure 2 breaks down the machinery behind this global tokenization. At its heart, the system is a diffusion autoencoder that maps raw protein coordinates into a sequence of discrete tokens. A key trick here is token dropout, which forces the encoder to pack the most critical global information into the earliest tokens. Then, for generation, an autoregressive transformer predicts these tokens, while the decoder reconstructs the final 3D coordinates using flow matching. This design effectively separates the token sequence length from the protein's physical size, enabling flexible applications like compression and protein shrinking. Well, following that architecture, Figure 3 demonstrates the practical result: how reconstruction quality evolves as we increase the information budget. The columns here display proteins reconstructed with 16, 64, and 128 tokens, compared to the original structure on the far right. What is impressive is the leftmost column. Even with just 16 tokens, the major secondary structures, those purple helices and yellow sheets, are already correctly positioned. This confirms that the model effectively prioritizes global geometry first, using later tokens to fill in finer details like disordered loops. Alright, having seen the visual proof, Table 1 now lays out the quantitative comparison against state-of-the-art models like ESM3 and Kanzi. Across three major datasets, Cath, Cameo, and AFDB, the full model, simply labeled hours, achieves remarkably low RMSD scores, hitting 0.90 angstroms on Cath. This effectively matches or beats the best baselines. The bottom two rows also track the 128 token versions. While the error naturally rises slightly when we compress the representation, the structural integrity remains strong, confirming the model's efficiency. So, Figure 4 expands on that compression idea by plotting reconstruction quality against the number of tokens. The three panels track distinct metrics: RFID on the left, RMSD in the center, and TM score on the right. In each case, the purple line represents the adaptive model's performance as we add more global tokens. Specifically, in the middle graph, notice how the error curve descends sharply. It crosses the orange DPLM2 baseline between 64 and 128 tokens, eventually converging with the high-performance ESM3 model at the bottom. This trajectory confirms that the model efficiently scales from a coarse approximation to a highly detailed structure. Well, beyond just reconstructing shapes, Figure 5 asks if these tokens capture meaningful biological properties, specifically for Cath classification. The left column shows the performance using an MLP probe, which is a small nonlinear classifier. Here, the purple line rises sharply as token count increases, eventually surpassing the dashed baselines for DPLM and ESM3. This indicates rich information content. However, looking at the linear probe on the right, the performance remains flat and low. The authors attribute this to the non-equivariant nature of the encoder, which entangles pose and structure, meaning you need that nonlinear MLP layer to unlock the data. Okay, while Figure 5 focused on classification, Table 4 proves the model can actually generate high-quality proteins. Here, we compare the autoregressive model against top baselines like DPLM2 and ESM3. The key metric is designability in the first column, which measures if a generated protein can physically fold. The proposed method, labeled hours at the bottom, achieves a massive 0.87 score. This significantly outperforms standard discrete models, which hover around 0.5 and even beats the best of N sampling from Kanzi. Wrapping up, two takeaways stand out. First, global tokenization effectively solves the error accumulation issues that limit local methods. Second, by decoupling token count from protein size, This approach enables novel zero-shot applications, like protein shrinking, while drastically boosting generative performance. That's it for this episode of the AI Research Roundup. I'm Alex. Thanks for listening.